No, no.
No?
It's my pleasure, Ravi.
Okay.
Thank you so much.
So thanks so much for coming.
So I just have to finish that anecdote. So Ravi, and go back to 1997. You wouldn't, I mean, look at the man. He looks like a... but nevertheless. So the first time, perhaps the time we met back then, there was a tiny little startup that some of you might know. And there was a mutual friend of ours, Shrikant, if you remember, who was trying to convince me that maybe I should consider working at the startup. Now, I should preface this by saying I have no memory at all. I cannot remember anything. So I don't prepare for talks because I wouldn't remember anything anyway. But over 20 years, the one thing I remembered was my meeting with Ravi. So he used to run a group called Mapping back in those days. I don't know if you remember, Ravi. And his job was, of course, the startup was Akamai, and now, of course, it's worth is it worth 100 billion now? No. Okay, tens of billions, yes. So this is a tremendously large company that Ravi was a big part of back in those days. And he used to do essentially measurements on the network, as in how much latency are you hitting, what is your pings and things like that. The funny thing is, now if you follow work in networking, a lot of this comes back in software-defined networking, where all of the work goes back to the kind of stuff that you guys were doing. And this stayed with me for 20 years. Amazing. So anyway, thank you so much for having me here. It's a pleasure to be here. Honestly, I do not know my audience, so I will take a stab at trying to do the best of all right, so I'll try to do the best of what I can with what I know of the audience. So Ravi said, don't make it about any specific results, make it about philosophy, open questions, things like that. So I will, there are maybe 7, 8, 9 papers worth of results somehow hidden in there, but I will skim over all of those and I'll try to extract out really what drives how we go about doing the kind of work that we do. And essentially this is, I call it the role of analytical statistical modeling in analytics. And of course, everybody is hard on machine learning and data mining, what have you. And so when we do this, we sort of take a somewhat unique tack to these problems. And I'll highlight what these problems might be, what our approach is. So just to make sure that we're all on the same page with respect to terminology, I'll just spend a couple of minutes talking about basic concepts, just a couple of concepts, nothing significant. I'll talk a fair deal about the kind of things that you probably know very well and what goes wrong in the kind of things that you know very well and trust. And then I'll motivate analytical frameworks. And really much of what I'll talk about is the meat of it is in part 2 of the talk, which is the statistical significance of what you might see as clusters in networks, the statistical significance of overlaps of what you might see in clusters, and their lineage. Let's say you're trying to track where somebody actually caught a disease. How would you actually track where this person caught that disease from? Something to do with the need for non-parametric priors. And the role that they might have to play in these things. And I will conclude with a lot of open problems. I really like simple things, and there are a number of very simple things that I do not know how to do. So I'll put these up there, and I'm sure there are wonderfully smart people here who can help me solve these problems. All right. So there are only 2 or 3 things that I will use as basic concepts. The first one is called a p-value, and I'm sure most of you know what a p-value is. And this is the probability of obtaining at least as extreme results, given that the null hypothesis is true. By the way, this is from the wiki, and it's a very nice definition, yes. So stated otherwise, this is a probability that a random selection, the second sentence is not from the wiki, that a random selection, null hypothesis, yields an observation as extreme or more, okay? So it's a relatively young crowd here. We have 30 people in this room. Perhaps all 30 of you are under 40 years of age. Now the p-value of this observation is the probability that a randomly drawn sample of 30 people from the general population contains people who are no more than 40 years old. It turns out that p-value will probably be quite low. And that says something about this population, which is that for whatever reason, there's statistically significantly young people sitting in this room. It also happens that all of you are probably computer scientists or mathematicians. which probably says something about the correlation between young people sitting in this room and them being mathematicians or computer scientists. We'll get to that later, okay? So we understand what a p-value is. A p-value simply is the surprisingness of a selection, okay? We have selected 30 of you here, you're all under 40. That is very surprising, okay? And the p-value of this is 10 to the power minus 4, okay? So the lower the p-value, the more surprising it is. And in some sense, the higher the statistical significance. Okay? But by the way, stop me, ask me questions at any time. Back at my home institution, we run a seminar called Curious Minds, and I don't think in 5-10 years of us running that seminar, anybody's gotten past slide 5 or 10. So just stop me. I don't claim to be right about most things, so at worst, I will admit that I'm wrong and you're right. So let's have fun. All right, so statistical significance is attained when a p-value is less than a prescribed level. Okay, so you're looking for low statistical significance. Now, it turns out statisticians use these for entirely different purposes. They use these for hypothesis testing, okay? In other words, that they will come up with a hypothesis, and they will have a bunch of facts, and they will estimate the p-value of those facts with respect to the hypothesis. And if the p-values are very low, then you reject the hypothesis, okay? So they're not using these to test the surprisingness of an observation. Rather, they're using these to test the validity of a hypothesis. So we're going to do this backwards. We're going to say that we're going to assume that the hypothesis is correct, and we're going to estimate the significance, and we're going to try and argue that the significance tells us something about what we're looking for, okay? So I'll make some blanket statements to begin with. Whenever you're trying to find something from data or observations, your data is always incomplete, okay? I don't care how densely you sample anything. You can know everything about my Netflix preferences, okay? I don't even know my complete sampling, okay? So that data is certainly incomplete. All data is incomplete. I will say nothing that you learn is absolute. In other words, you can formulate the nicest convex optimization problem, you can solve using your nicest methods. It doesn't matter what the answer is, because it's working on data that's fundamentally incomplete anyway. So no answer is absolute, all data is incomplete. And typically in the real world, you will never be able to get the absolute answer anyway. Nothing is convex. And when you're looking at these problems, most of them will be some awful optimization problems that you can at best say, this is a solution, not the solution. Okay? We will look at examples of all of these things. All right. So let's start simple. I'm sure most people in this room know what classification is. I'm going to pick on Ravi. Ravi is a connoisseur of fine coffee, and perhaps he's a coffee drinker. I could probably go through this room, and I could probably easily pick out people. And with the 90% precision call, I can classify you as a coffee drinker or not a coffee drinker. Okay? Should you be impressed by my prediction prowess? Well, what if I told you now that 90% of all people are coffee drinkers? Okay? So now my classifier is completely worthless. Okay? I could have called all of you coffee drinkers. We could have played this game, and I would have given you 90% precision recall. And of course, it would be completely meaningless because the null hypothesis says that 90% of all people are coffee drinkers. Okay? So any measure of things like precision and recall must be viewed in the context of the null hypothesis, okay, or the background distribution. If the background distribution is biased, then you can easily get good classification accuracy. All right. So what I'm trying to say here is, in the context of the null hypothesis, a classifier with the 90% precision recall has no statistical significance at all. So if you were to establish a p-value for that particular classifier, that p-value would actually be quite high, okay? And you should actually reject that classifier, okay? The p-value actually tells you even more than just whether a classifier is good or not. Let's say going from 90% to 91% was actually very high. A good p-value will actually be able to say that a 90% accurate classifier has a p-value of 10 to the power minus 1, whereas a 91% classifier has a p-value of 10 to the power minus 5. Okay? So it'll actually give you more than simply a point estimate. It'll actually give you sensitivity of what you're looking at as well. Okay? So that's one example of what a p-value does for you. All right. So this is something else perhaps all of you in this room know very well. This is called association rule mining or associations. This is the kind of stuff that people do when they say, somebody that buys bread also buys butter, okay? Or I think there's a famous example here, which apparently is an urban myth. It's actually not true. This is the Huggies and Heinies example, where somebody claims that people buying diapers after 5 p.m. tend to also buy beer. It's a very popular example, but I've actually heard that this is actually not true. But nevertheless, so people who do this, typically the algorithms that we know how to work on these works in two steps. In the first step, you find all frequently co-occurring items. And then in the second step, you just do a conditional probability on it. Okay? So how do we find frequently co-occurring items? You use something called, first of all, you use a measure of frequency as co-occurrence, which is terrible. And then you simply use downward closure, which is that every subset of a frequent set must itself be frequent. And therefore, you start with one sets, prune out everything that's not frequent, use the one sets to make candidate two sets, prune out everything that's not frequent in candidate two sets, and things like that. Okay? So this is the state of the art. I mean, people have made their careers doing stuff like this. Well, turns out, if you think about it, it does not work very well at all. Frequency is no measure of statistical significance. Okay? So if 1% of the people bought bread and 1% of people bought butter, in fact, if you maintain the same statistical significance, even under the weakest priors, which is IID, everything is bought independently, everything has the same probability, which that is a very weak assumption anyway. Even under this very weak assumption, you cannot keep the frequency the same. So if 1% of the people now bought bread and butter together, the statistical significance of that two set is much higher than the statistical significance of the constituent bread and butter sets. Okay? So frequency is a terrible, terrible measure of statistical significance. The consequence of this is that what comes out of these things is actually the prior. You will never learn anything meaningful out of this. If you wanted something like anchovies implies pizza sauce, you will never find it out of this because anchovies never rises to the level of statistical significance for anything. This will tell you, yes, bread implies butter, but then you throw up your hands and say, well, tell me something I didn't already know. Okay? Then what they'll do is, people who do this kind of business, they have this notion of support. Remember the frequency I told you? They will lower the frequency, and once you lower the frequency, you now have millions and millions of these rules popping up, which then make no sense either. And then they'll come and say, well, let me now give you heuristics to prune those rules. All of that happens because these people never figured out how to find statistically significant sets to begin with. Turns out that's a much harder problem, okay? Because these notions of downward closure, monotonicity just don't work. If I were to simply say, here's a set of sets, tell me everything that occurs statistically significantly together, even under the weakest priors, we do not know how to solve this problem, okay? But nevertheless, this doesn't work. So it is a terrible way of solving this problem, but nevertheless, this is one of the cornerstones of what people do in data mining. All right, this is even simpler. I just give you a bunch of uniformly distributed points, and I give it to k-means. What happens? It'll give you k-means. This is stupid. There were no clusters to begin with. So you have to tell me what the significance of your answer is. I actually don't even care about the answer. You start by telling me whether an observation is statistically significant or not. So what I'm trying to tell you here is the algorithm itself should actually be an afterthought. Perhaps what's even more important than the algorithm for finding an observation should be what is the significance of the observation, okay? And establishing the significance of the observation is actually a very hard problem. You have to go through how to find an appropriate prior. You have to go through how, given that prior, you're going to characterize the statistical significance, which gets us into all kinds of really nasty analytics, or the average case behavior of these things. If you look at Monte Carlo or sampling type methods, nothing converges in typical problems. So all of these become extremely hard problems. Now, once you get past all of these things, what am I really looking for? I'm looking for very low p-value events, okay? The way I learned all of these things is I dabble a bit in biology. So in fact, a lot of this talk is going to draw fairly heavily from systems biology and maybe even some biophysics. But I won't cover any of that. You'll just see snippets of that. Ask me a question, or I'll direct you to a bunch of papers. So I learned about these things really from things like BLAST. I don't know how many of you use BLAST to line up sequences, okay? So when you line up two sequences using something called BLAST, which is a tool that everybody uses, it gives you two things in addition to the alignment. It gives you a p-value and an e-value, okay? The p-value, of course, in biology, everybody says until you hit a p-value of 10 to the power minus 5, the match is not significant, okay? And people will realize that even more important than the Smith-Waterman or FASTA or whatever algorithm you use to match these things, even more important than that is the p-value of the match. Then why not set up an algorithm that explicitly minimizes the p-value? Because clearly we're looking for the minimum p-value. Well, turns out none of the algorithms we know actually are proven to minimize a p-value. So yes, in fact, we can formulate this as a longest common subsequence or what have you, and then take the sequences that come out and use another procedure to then establish a p-value for these things and say, aha, what came out of the LCS problem has a p-value of 10 to the power minus 6, and therefore that's not a random match. I should be excited about this. Please. Sure. I'll get into a lot of that. At this point, I'm talking in abstract. Yes, please.
Well, I guess I'm just confused about whether the quantity itself is the best fit according to the structured model, or whether it is a bit of a particular, fit that I found.
Beautiful. So that's exactly the point I'm trying to make. So what you're saying is the longest common subsequence, which was some abstraction or approximation to what I thought was the model, gave me the match, okay? Then I fed this match into this other black box, which gave me the p-value, okay? So in an ideal world, my algorithm that I was using really should be minimizing whatever this black box is using to compute the p-value. But it doesn't. In almost no cases does it do that. Turns out that's a very hard problem in most cases. Turns out, actually, we can't even take a step backwards where I can give you a bunch of clusters in two-dimensional, I can give you a point set with clusters in them, and I ask you to put a p-value on it. It turns out that's not a trivial problem. I can give you a weighted graph, and I can say, find me the most statistically significant cluster in this weighted graph. I do not know the answer, okay? So I do not know the answer both ways. I do not know the algorithm for minimizing the p-value, and I actually do not know how to establish a p-value for an observed cluster. Okay, so I will mostly focus this talk on establishing a p-value. And I'm going to say, fine, we separate the algorithm from the p-value computation itself, but that there's value to both of these. Please, Deeparnab.
How do these iPods establish a p-value?
So I'll talk about, at least in the context of graphs, I'll talk about how do you establish a p-value of a cluster, how do you establish a p-value of the overlap of two clusters, few other examples that I'll go through. Please.
In many, like case in biology, we find a motif in a network.
Yes.
The thing is to see whether it's significant, you randomize, keeping the degree and everything.
Sure.
One hypothesis is that is the motif simply because of the degree distribution.
Sure.
Checking with them.
So that's a good example. So motifs are, just to put a little more context on the question, there was a fairly influential paper by Yuri alone about, I think, 7-8 years back. It was in Nature or Science or something. So what Yuri alone says that things like feed-forward loops, feedback loops, triangles are statistically significant. He still uses frequency as a measure of significance. Okay? So he will first say, I found an overabundance of triangles that go like this, or feed-forward loops which go like this, and then he argues one is a stable point, one is a delay signal, things like that. But even their measure is statistical significance, and the way they find it is once again by permuting edges and doing Monte Carlo samples on these things. And as I'll briefly talk about it later, these are exponential spaces. So when you're sampling in exponential spaces, you have to sample a great deal before you get to convergence, okay? So there are all kinds of problems there. But there are actually, with that alone paper, there are other problems as well. There's nothing sacred about 3-edge feedback loops. There could be 4-edge feedback loops. So the motif should be a lot more general. So yes, it's exactly along the lines of what I'm... Exactly, yes. I'll get into all of that, please.
Algorithms which will minimize p-value. I'm thinking about a situation, suppose I have some data points and I need to look that if there is a cluster
Absolutely.
or not.
Yes.
So if we go for something where the algorithm itself minimizes the p-value, are you not approaching to overfit the data? Because the thing is that the algorithm...
Okay, hold on. So the point you're making is, what is the p-value computed with respect to, okay?
Yeah.
So the p-value is computed with respect to a model, not with respect to data, okay? So for example, I've given you points in two-dimensional space, okay? And I've said that these points are drawn from a mixture model, and the mixture model is two Gaussians, let's say. Okay? Now, I'm taking this point set, and I'm computing the best clustering of these point sets, which is the lowest p-value with respect to the prior, not with respect to the data. Okay? And that's why the design of the prior is so important. Okay? So you'll never overfit to the data. You may overfit to the prior, but not to the data. Okay? So you have to believe that the prior is right. And I will talk a little bit more about what a right prior is. All right. So these are some questions that I threw out, and I'll answer these questions. So let's say you run graph clustering on Facebook, and you find a group of 300 people who are almost completely connected. Should you get excited about this? Turns out you should get excited about this. I'll give you some analysis which shows that if you put in all the parameters of Facebook, the expected size, at least in an Erdos-Renier graph, of a row-dense component with a p-value threshold of 10 to the power minus 3 or something, is about 140. So if you find more than 140 people who are mostly connected with each other, that is very surprising. Okay? And I will give you an analytic on how you do that. Okay? Here's another example. 70% of your, I apologize for any stereotypes in here, but 70% of your friends on Facebook are male, 60% of your friends are computer scientists, 80% of your friends are Indian. Do these numbers and their overlaps tell you anything? Maybe it tells you that you know, most computer scientists are Indian and perhaps male. Okay? But if I'm just, once again, I apologize for the stereotypes, but to the extent that this data is what you're seeing, is that the hypotheses that you draw from this data? Okay? And I will try to answer this question as well. Not in this context, but in the context of something called ego nets and stuff like that. All right. Here's another example. 75% of everyone's friends on Facebook live within 50 miles of them. We have data like this, not on Facebook, but on some other social networks. Now, can I use distance as an indicator of friendship? Okay? So this is topology completion. There's the converse of this, which is geometry completion. Okay? Well, it turns out the implication goes one way, but not the other. In other words, if somebody is your friend, more than likely they are within 50 miles of you. But really, if somebody is within 50 miles of you, it doesn't mean that they're likely to be your friend. Okay? So you can use p-values to establish all of these things cleanly. And finally, there's something that I will pose as a question, which I have some answers to. And this is, you know, can I trace the flow of a contagion through a network? It's lineage in the form of a DAG, perhaps. Okay? And I'll talk about all of these answers. Okay. So in each of these questions, I will argue I'm looking for surprisingness of the observation. In other words, I'm trying to find observations that minimize p-value. Turns out there are other classes of problems as well. Here, you can look to maximize the probability with respect to a prior. Okay? So for example, given a dynamic network, and once again, I'll give you some answers to these questions. Given a dynamic network, who was the first guy on the network? Okay, so this is Facebook. Everybody's deleted their join dates, and you're trying to find out who joined Facebook first. Okay, or you're looking at a protein interaction network, and you're trying to find the ancient proteins, the prokaryotic proteins in the network, things like that. Now, of course, what you will do here is to then have a model for the dynamic evolution of the network. This could be a preferential attachment model. And then you're saying with respect to preferential attachment model, what is the most likely node arrival sequence? Okay? So that's the point I'm trying to make here. Now, in each of these questions, we're looking for an explanation most consistent with our understanding of the driving processes or priors. And these would maximize the probability with respect to the prior. So in other words, here we're trying to construct a solution that maximizes probability. In the previous case, we were trying to minimize the p-value. And we'll look at these. All right. So what kind of problems are we looking to solve? We're typically looking to solve these problems which get posed as optimization problems. Okay? Turns out the optimal is typically infeasible at scale. It's often an overkill because of noise and missing data. But most people still do this. This is what the state of the art is. The obvious solution is, the solution is one that you get from most analytics algorithms. However, this is rarely useful. The most statistically significant solution is one that poses statistical significance as the optimization criteria. It turns out this is just as hard to do. Now, the one in the middle which says any solution that has statistical significance really is the one that most people look at, or at least most people, when they have to quantify their answers, would be looking at. Essentially, this trades off feasibility and utility. In most cases, this is a solution of choice. You use a heuristic to find an object, an artifact, and then you use a black box to establish a p-value for the artifact. And if the p-value is low enough, you can argue both the method that you used as well as the observation. Okay? Anyway, so let's look at some examples now. So, just once again, to recap some of the points that have been made already, p-value can be formulated in different ways. For example, formulating a p-value of Facebook, you can ignore the graph and simply look at its hypergeometric p-value, okay? It turns out that in most cases when you're dealing with things like graphs, losing structure is terrible, okay? So if I were to take a graph, ignore the graph structure, just treat it as a hypergeometric, you're going to make mistakes. So you have to formulate the p-value properly. Desirable p-value formulations provide discriminating power. In other words, that if there's an observation that is significant, an observation that is not significant, one should come out as low p-value and the other one should come out as high p-value. In graphs, most of the times you will notice that if you don't design your priors well, if you don't design your p-values well, you get very sharp cutoffs. Everything will come out as being statistically significant. Take Facebook and you model it as an Erdős-Rényi, and you're looking for cliques in Erdős-Rényi, everything that you'll find in Facebook will have a 0 p-value, because you don't find cliques of that size in Erdős-Rényi, okay? So designing the p-value, designing the priors are all critical. Priors may themselves be non-parametric. So very often when you're doing these refined analyses, you cannot use things like power law or Erdős-Rényi or any of these models as priors anyway. And p-values may be analytical. These are often very hard to derive. I find them very hard to derive. Smarter people probably not so much. And they might be empirical, which is the URI alone kind of way of doing it. It turns out these are often very expensive to compute because of the number of trials required, especially when we're talking about graphs. All right, so let me start with the first example. This is the statistical significance of clusters. And the question here is as follows. You run graph clustering on Facebook. You run mCode or something like that. And this finds you 300 people who are all almost completely connected. Should you get excited about this result? Mathematically stated, the question is, what is the significance of a dense component in a network? And you can generalize this also. If I give you two networks and you align those two networks, you could ask the question, what is the statistical significance of a conserved component in multiple networks? So it generalizes fairly easily because you can take the two networks, compute the product graph of those two networks, and the alignment shows up as a dense component in the product graph. So these two questions are exactly the same, and they're the question that we talked about before. All right. So I'm going to do this mostly in protein networks because this is where we prove the results. It turns out most of these networks exhibit power law type properties, exponential geometric things like that. Analysis typically is simplified through an independence assumption. And it turns out that independence assumption may cause problems for networks with arbitrary degree distribution. But nevertheless, that's the only analysis I know how to do. And I can essentially model this as a GNP or an Erdős-Rényi, which essentially means that you have n nodes in the network. The probability of any pair of nodes being connected is p. And you can simply compute p from the data that's given to you. So I've given you Facebook, and you know that the average person on Facebook has about, I think this is a number that keeps increasing. When I had the data, it was about 140 friends. But I suspect now it's much more than 140 friends, which means that the average node degree is 140, and you can use that to compute p of a GNP. And so now we essentially have a GNP that serves as the prior for this cluster question that I talked about. I'll give you the results for the GNP, and I'll also give you the result for an extension to what we call a piecewise GNP, where there's a GNP at the bottom and a GNP at the top. P is higher here, P is lower here, and the connectivity is parameterized between these two. The reason I do this is because I don't know how to do this for power law, and really our networks were starting to look more and more like power law, and I said, okay, the next best thing I can do is piecewise GNP. I don't think the answer exists for power law even up until now, but I actually conjecture that asymptotically it is the same answer. All right, so let me just define a row-dense subgraph. This is just a subgraph which has got a fraction row of a clique, okay? So a clique has order n square or n square over 2 edges, and I multiply that by row, which is somewhere between 0 and 1, and that is how I define a little bit of sparsity inside of a clique, okay? So the question to be answered is, what is the expected size of the largest row-dense subgraph in a random graph? Okay? And any row-dense subgraph with larger size is statistically significant. Turns out this is actually fairly icky analysis. I'll just speed through this a little bit. Actually, I won't do the analysis for you, but the takeaway from this is that the expected size of a row-dense subgraph in a GNP is roughly log n. Okay, and there's all kinds of constants sitting in there, but let's not worry about that. In this paper, this is about 10, 12 years back, we thought we were the first ones to show this result and another result. Turned out there was somebody else who'd done this about 5 years before us, and that's terrible because we didn't cite that person. Turned out somebody else proved this result again in 2011 without citing us, and turns out somebody else did this again in 2013 without citing any of the others. But nevertheless, I mean, I don't feel so terrible because we did have the second result as well, which generalizes this result. Anyway, so the bottom line is this. You expect to see a row-dense component in a GNP network of roughly size log n, but then we actually give you the first order moments and a complete characterization, so you can actually get the entire distribution, which means that if you give me any size of a clique, I can immediately tell you, okay, the p-value here is 10 to the power minus 10, okay? So we completely characterize the p-value for this. We actually also generalize this. Oh, so this was the answer. Actually, I might have been wrong. So plugging in the values of NP, setting rho equals 0.9, the p-value of a 300-person row-dense component in Facebook, picking all the numbers from Facebook, is about 10 to the power minus 4, okay? So it is actually very surprising. You will not find, randomness will not cause you to find a cluster of 300 nodes here. Please. Assuming that the null model is GNP, yes. So I would always say that in our experience, like I said, we do a lot of this mostly in biology where either something is correct or not. The question to ask is, can you get classification out of this? So if a GNP always gives you p-values which are very low or very high, that's typically what happens, then it immediately tells you that the prior is no good. Okay? But it turns out that if you reduce the size there from 300 down to something else, you actually get very nice classification. Okay? You don't want your prior to be exactly the same as data anyway, because if the prior is exactly the same as data, almost nothing shows up as surprising. You don't want the prior to be too far from the data either, because everything becomes surprising. So it turns out that this actually does give you, I didn't actually run the entire characterization for Facebook, but I will give you examples for the protein interaction networks that we talked about before. So the point is wonderfully well taken, that these networks are not GNP. Clearly, we know these networks are not GNP. So what can we do? So the next result is essentially that two-level GNP model that I talked about before, okay? So you have high connectivity nodes at the top, you have low connectivity nodes at the bottom, and some connectivity between these two, and there's a whole lot of miserable math that goes into it to essentially show that the expected size of the clique is also log n with an additive constant that we can show, okay? So we can fully characterize the distribution of cliques inside of this two-level GNP model as well. I will conjecture at this point that this result will generalize also to power law distributions. In other words, that the size of the largest size of the expected size of a row-dense component in a power law model probably is going to be log n plus an additive constant that kind of looks like this. Okay, but these are very icky things to do. These are very painful derivations to compute. So what can I do with this? It turns out actually you can do some very easy and fun things with this. So come back to the problem of clustering graphs. I will take the graph that I started with, and I'll apply some min-cut heuristics. And I essentially terminate applying that heuristic when I get to a point where the p-value of these leaf clusters is below some statistical threshold. Okay? So I think you set this up as 10 to the power minus 5 or something, and you stop at that point, and now you can evaluate each of those clusters separately. It turns out that the state of the art is actually a very nice algorithm. I don't use my algorithm. I actually use MCode myself even now, in spite of my result here. Mostly because the min-cut algorithm that we were using is not a very robust min-cut algorithm. But nevertheless, the state of the art is MCode. If you ever want to do graph clustering, you should download this. It's open source, very nice piece of software. It turns out that in terms of specificity and sensitivity, you can beat MCode very easily using the p-value heuristics that we talked about before. Okay, you can actually, and of course there are some biological ways of estimating accuracy here. So we use something called gene ontology annotations to do this. Turns out if you look at all of the plots of size versus significance, sensitivity versus specificity, size versus specificity, and size versus sensitivity, you will notice that a simple heuristic that we talked about before quite handily beats out a very nice and established piece of code like M code. Please. As please.
So, first, I like this framework in general. But, suppose that I had a null model. I guess we're also calling it prior. Suppose I have a null model in which the way that Facebook is generated is that there are clumps of 400 friends,
Yeah.
which are dense and
Yes.
very, very sparse connection.
Yeah.
So, if you now find a clump of 400 friends, this is not surprising.
Absolutely.
On the other hand, it doesn't mean that you're wrong that that's a real clump or that that's the clump to which those people belong.
No, no.
So, is the goal to classify graphs as in this is a clumpy graph or it's not? Or is the goal to say, yes, you found a real clump?
So in your example, when you said that my prior is, clumpy 400 node graphs, so the question you're asking is there anything in this data that I didn't already know? Okay? So
You didn't know who belonged to which.
okay, that's a different question. And there you can get into other priors of who belongs to which clump, if that was the question you were looking to answer. If your prior was the structure of my network is clumpy with the average clump size of 400, and if you got nothing with low p-value, essentially you will come to the conclusion that there's nothing in this data that is surprising to me.
Okay, so you're not addressing, you're addressing a different question. So and I thought but you're not addressing the question of when do I know that a clump in a graph is a real clump? You're addressing the question of when do I know that Facebook has the property that it forms clumps?
Okay, now you're taking the hypothesis testing point of view, you're taking the statistician's point of view. Now my point of view is this. Given that you have a certain understanding of data, okay, you know that the data is generated by these clumpy generation models that you've come up with. Is there something surprising in Facebook? That's the question I'm trying to answer. Okay? I'll give you a few more examples and we'll come back to that question. Maybe the other examples will answer this. All right, so let's get to, so now we know a little bit about how to find significant graphs, okay? By the way, this is actually very important. We've spent maybe the past 15 years really closely analyzing all kinds of data in biology, everything from single cell expressions to interactions to dynamics of interactions. And really the most reliable way of validating our answers is this framework. So it really, I don't say it lightly, I really do believe that at this point, this is really the best way of validating our answers. Especially when, I joke that the worst thing that can happen if you make a wrong recommendation on Facebook, on Netflix is that the person doesn't watch the movie. But when you're trying to say that this molecule is a drug and that gene is a drug target, you do not have that luxury of saying, too bad, I screwed up. You have to have a p-value of 10 to the power minus 200, that gene is causing something, or that set of mutations is causing a certain phenotype, and that drug molecule is going to knock down that gene. Okay? So when you get to more refined analysis, this framework almost is critical. It's worked very nicely for us. All right, so let me now go to the next problem, which is I know how to find clusters, and now I'm finding multiple clusters, but some of them tend to be overlapped. When is the significance of this overlap? How do I characterize the significance of this overlap? So this was the problem we started with. 70% of your friends, God, I gotta change this example. Okay, let's change this. 70% of your friends on Facebook are female, 60% of your friends are mathematicians, and 80% of your friends are from Burkina Faso. Do these numbers and their overlaps tell you anything? Okay, so let's look at that. Now, the first thing when you design a p-value is you have to ask yourself what is surprising to you. So I say that an overlap is significant or surprising if it is at least as dense as the constituting subgraphs, okay, and it is large enough. Okay, so I have some intuitive feel for when I expect this overlap to be surprising. All right, so based on this, I'm going to define something called the p-value, and I call it a CoDO p-value. It's just a p-value that we come up with. Essentially, it's a p-value that is conditioned on the size of the two clusters, the density of the two clusters, the size of the overlap, and the density of the overlap. Okay, so once you condition the probability on all of these things, you actually get a robust p-value. Okay, so if you go past the formalism here, essentially that's what p-CoDO does. It's something which says that the overlap of these two must be greater than a certain size, and that the density should be greater than a certain density. Okay, and I'm assuming that A and B are the clusters are themselves dense. All right. So by conditioning on the size of the overlap, you can actually get an explicit formula for this in terms of hypergeometric tails, and I'm not going to go into the details. This is really nasty, icky stuff. I'm not a statistician. I'm barely even a computer scientist. On a good day, maybe I am. But nevertheless, this is a level of statistics that I do not enjoy. But nevertheless, we can find a closed form, and then we can actually start to do fun things with it. So here is an example. Here are 4 different examples, actually. So I have a blue cluster and a green cluster and a set of red dots in the middle. And the question I'm asking is, are those red dots statistically significant? Okay, I've defined a bunch of p-values here. There's something called HGT, which is simply hypergeometric. Defined something called ERD, for which I'm simply looking at the overlap subgraph, and I'm characterizing the statistical significance of just that little overlap part using the result that I had before. And the third one is this p-value that I've shown you, the CoDO p-value. Okay, and you will see that in all cases, CoDO is actually both rigorous in the sense when the overlap region becomes small but the density is large, we can still find it. When the overlap region becomes large and the density is okay, we can still find it. And all of the other methods fail in different parts of the regime. Okay? You can also show that this p-value is actually a very smooth transition. So it actually lets you discriminate something that should be tagged as insignificant from something that is tagged as significant. So let me give you an example of where this works. So we compute something called the EgoNet. This is the induced subgraph among friends. This is, I think, Kleinberg's terminology, so I take no credit for this. So this is, your friends are your alters, and you are the ego. So you build these EgoNets, and you can build these EgoNets for Facebook, Google, and Twitter, and you will find much better characterization of the overlap between the EgoNets using these CoDO p-values that I talked about, as opposed to a vanilla hypergeometric or simply characterizing the significance of the overlap set. You can actually do much more fun things with it. So here is, I don't know how many of you follow systems biology, but KEGG is sort of a fairly standard database of networks of pathways. These are how cells perform different functions. And the question is, what is common to these pathways? Okay, so you're using p-values to characterize overlaps of these pathways. And the edges here essentially signify the overlap. So think of it this way. This is a pathway, this is a pathway, and an edge exists when there's a significant overlap between those pathways. Okay, so all I've done is I've just computed the CoDO p-values on all pathways inside of KEGG, and out pops an overlap network that looks like this. And it turns out you can actually find the dense parts of this. So this is all the pathways that have to do with drug metabolism. The ones in the blue are cancer signaling pathways, immune response. That one is not as dense, but nevertheless, we know that those sets of pathways do DNA replication and repair. So you can actually find, quite by magic, these really neat associations between pathways simply by assessing the p-values of overlaps of those subnetworks. All right. So let's look at another example. This one I won't give you a whole solution to. It's a very simple problem. So the problem is as follows. I found 2 clusters, A and B, and you find an overlap between those 2 clusters. So this is, the clusters themselves are A and B, but I've called them 3 parts, X, Z, and Y. Okay? And the question is, how should you install this in sort of a tree or a DAG? Okay? So most of us do some kind of hierarchical clustering. And if you had hierarchical clustering, what would you do here? Would you just take the root node and have 2 clusters, A and B? How do you recognize the fact that A and B actually have a fairly significant overlap between them? okay? And I'll try to answer why this problem is, by the way, I hope you recognize the difficulty of this problem. I've spent the past 2 or 3 months looking at this problem. It turns out this problem is really, really hard, just even at those 2 levels. So here's what we've come up with. Please.
So, but I'm not sure what you mean by a good answer to this question. I mean, by lineage, do you mean some causality or?
How these clusters might have evolved. I'll tell you exactly where these are from. So in biology, there's these really new technologies. These are called single cell technologies. And so we can actually look at how genes are expressed in a single cell. Okay? So what you get are these long vectors. These are expressed genes in single cells. And so we're trying to push this, we and a few others are trying to push this new hypothesis that up until now, people have believed that lineages of cells are hierarchies. In other words, that you start from something, you specialize into 3 different types of cells. You know, start at the embryonic stem cell, you get 3 differentiated cell types, and these differentiated cell types become the precursor cells, and you get a nice little tree of these things. But we've been trying to argue that it's actually not a tree, but it's actually a DAG. And there's some evidence to suggest, and certainly, you know, you look at the Yamanaka things, the reprogramming of stem cells and things like that, that even in late stages, you can reprogram a stem cell to look like something else. So you can have one cell that specializes from multiple lineages, okay? So now think of these groups as essentially these expression profiles. So I've taken a bunch of expression profiles of some cells, a bunch of other expression profiles, I've clustered them somehow, and this is what I'm seeing. Okay? And I'm now trying to infer from these, how did those cells specialize from what? So I'm trying to build either a tree or a DAG on top of this. Of course, I have to come back and show you that a DAG is better than a tree. I'll give you some examples of what that looks like, but that's sort of where I'm coming from. You could also see this as, in the context of this workshop, there's a bunch of people who've caught a disease, and there's a bunch of people who have the same disease, but if you cluster them, you see this overlapping cluster. Okay, and the question is, what was patient 0? What was the site from which these diseases came about? In other words, you know, if this was Bangalore, this was Mysore, and this was someplace in between, how did that disease come about? Was it one person who gave it to those 2, and they evolved independently, or that were there actually 3 groups that evolved from the root? Okay, so those are the kind of questions we're looking at. Turns out these questions are actually very hard to answer rigorously. So first of all, we need to make sure that all of these clusters, X, Y, Z, are statistically significant themselves. We need to make sure that one of the clusters is not contained in the other. So you have to define some notion of statistical significance of containment. We do this using edge cuts. And you need to assess the statistical significance of the overlap of these 2 clusters. It may be that that overlap part is just noise. Okay? If that overlap part is noise, then you can clearly argue there's a root node with 2 children, A and B. If that overlap starts to become larger and larger, that's not noise. There's a different model that explains how those clusters came about. Okay? So these are the kind of questions we need to answer. And once you've done everything right, you can then argue for any of these based on the various statistical significance measures that you've defined. You have something where this is a DAG. You can have something where these are completely independent paths, or you can have something where these are derived from the root, things like that. So based on those p-values, one of these is actually the right instantiation of that very simple overlapping cluster that you saw. And there's about 6 p-values that go into computing this. So it actually becomes a very, very hairy problem. So when you do all of this right, you actually get something like this, and this is beautiful. So this is at the sort of the cutting edge of biology. We understand no biology. I have, beyond my high school biology, I've never taken any biology. But it turns out that if you do everything right, this is what we derive, and this is actually very nicely substantiated. So there was a recent paper out of Ellen Rothberg's group at Caltech, which draws an experimentally derived lineage of hematopoietic stem cells, which actually looks shockingly similar to this. And of course, we were overjoyed when we saw that because we had no way how to validate this. And that was sort of our first validation step for this. So if you do a Google on Ellen Rothenberg review in Nature Communications, you will see something very similar in that experimentally derived graph as well. And like I said, there are so many variables here. If you don't do this statistically rigorously, you will never catch the right signal. That's what I'm trying to, sort of the take-home point of this talk. Please.
So, you take a Bayesian view, like David
Jensen's
Yes.
paper model, right?
Yes.
Which learns these tags, which
Yeah, so there is a notion of an implicit prior in those things, and there is a notion of a p-value that's hidden deep down somewhere in there. I've never been able to actually characterize what that is. So they take an alternate view to exactly the same problem, but I would argue that there's deep down in there's a p-value that they're trying to minimize. Of course, they won't do it exactly either, but you can reduce David Jensen's model sort of to this high-level framework. All right, so up until now, I've mostly messed with parametric priors. Turns out parametric priors don't always work. So here's a simple problem. So I've now given you about 79 different networks. These are 79 different things that happen in 79 different tissue types in your body. And I don't know if you know, but biologists like to use yeast as a model organism for everything. And so the question you're asking is, does blood look like yeast? Does the biochemistry of blood look like yeast? Does the biochemistry of a neuron look like yeast? And you're doing that simply by trying to line up networks and assessing their p-values, okay? Well, it turns out here the problem becomes one of simply building a suitable prior. So the question you might be looking to answer, and I'll give you the results for these, is for which tissues is yeast a good model organism? And once you've done that, what are the shared missing functional components in yeast compared to human tissues, okay? Here it turns out that if you look at all of the 79 different networks, you're trying to align 79 different networks with one, and you're trying to say which ones align well, which ones don't align well. Turns out almost everything aligns well, because our body chemistry is actually well modeled by yeast for the most part. So you have a network of about 10,000 nodes here, 7,000 nodes here, line them up, you will have about 4,000 nodes that line up beautifully, okay? Now you will realize almost immediately, if this is the case, the p-value should not reward anything for the alignment of those onto the yeast interactome, okay? So in fact, here what we do, the first thing we do is that we take all 79 of those networks, we line them up against each other, and we actually build a network called the housekeeping network. This is the network that is common to all cells, okay? And just because any of this maps onto yeast should not give you any statistical significance at all, okay? So immediately I have the 0th order prior. The prior is the alignment of the tissues with each other. Then you build distributions of all the other nodes and the frequencies, and you then have a non-parametric prior. Please. Will it punish the data sets when it doesn't agree with?
You see that, the housekeeping genes are common.
Yeah.
Interactions are common.
Yes.
And you realize that you get a data set where the housekeeping genes are not overlapping at all.
Yeah, so actually so I'll give you some example of how to deal with this. What we do is we sort of, Okay, so we actually deal with it by taking this consensus approach that everything that's in the housekeeping, we sort of assume that it's going to line up anyway. So more than likely what happens is that when housekeeping genes don't line up, it's probably because data is dirty, not so much because there's a missing gene or two expressed here but not expressed there.
In principle, there's a scenario when there is no overlap in housekeeping.
Yeah.
That will be missed. But the point is, it happens very so rarely that you can.
So there are two things to this. The first thing is, how do you define housekeeping? Okay. So if something is expressed in 60% of human tissues, would you call it housekeeping or not? So there's one threshold there that you pick. And the second threshold is, once you pitch something as housekeeping, what do you do if it misses? And you can clean that out. We mostly don't care about housekeeping at all. You're right, if something is missing a housekeeping gene, it should cause a problem with the p-value. We do not believe it does, so we just ignore it. So now we can define a nice little p-value with respect to this non-parametric prior, but then there's no analytics here. So now in my lab, we have about 200 machines, and they run for a month. All they do is do alignments of these things. So if you do about 10,000 alignments with those random tissues with one tissue, you will get one p-value, which will be either 10 to the power -4 or less. Okay, so that's how much work it takes to do these things. But then when you get these things out, they look beautiful. So these are the most conserved subset of housekeeping genes. Turns out this is ribosome, proteasome. Those are some metabolism pathways, things like that. So these are all beautiful pathways that we all know should happen in every cell, okay? So I apologize for how small these writings are, but trust me, in the paper we make a very clean description of what these are. It absolutely validates our understanding of biology. But even more interesting, so those were the 5 big pieces. Even more interestingly, you can now look at all of those cells and say which one looks like yeast and which one doesn't. So these are all the ones that look like yeast, myeloids and monocytes and dendritic cells, NK cells, T helper cells. You look at the ones that do not look like yeast at all. Most of the nervous system cells do not look like yeast at all. And I'll show you the reason why it's not. So once you've done those alignments, and once again, if you get those p-values right, you can actually tell me why something looks like yeast and why something doesn't look like yeast. So this is what is conserved between blood and yeast, and this is what is human specific. In other words, you did not find this in yeast. Okay? So it turns out that the reason why blood cells are modeled so well by yeast is simply things like cell cycle. So blood is non-proliferating. It doesn't actually form tissues. There's no cell-cell signaling inside of blood. So all of these makes blood look a lot more like yeast. So things like DNA repair, DNA packaging, DNA replication, cell cycle, nuclear envelope organization, histone acetylation, things like that. These are very common. Okay? Now it's beautiful when you look at the brain tissues that I talked about. And the reason brain tissues don't look like yeast is organ development. That's some kind of a signaling pathway across neurons. Glutamate receptor signaling. So these are neuron-neuron signaling. There's no signaling across yeast. That's neuron-neuron transmission. None of these exist in yeast. So once again, just by getting your priors right and getting your p-values right, I can actually find, even somebody like me who doesn't understand any biology can find you these really neat things that make so much sense in why these networks do or do not look like each other. All right, actually, I'll skip some of these parts, but you can actually go deeper down and say, once you've found these brain-specific networks, can you look for genes associated with disease? So here, we're talking about diseases such as immune or infection or psychiatric disease or neurological diseases. And what I've shown you is that the p-values of finding these things are much lower in the brain-specific parts of the network than the housekeeping parts of the network. So what this tells you is that if you're looking for druggable molecules, you will more than likely find them in those brain-specific parts of the network. So you can actually do very nice things with these p-values. I'll skip some of these examples. All right, so let me end this with some things that I don't know how to do. And these are incredibly simple things. So your favorite search engine, I should not admit how I know this. They typically search for keywords, and they use proximity of keywords as one of the primary heuristics for ordering search results. Okay? In other words, if you're looking for average case analysis of algorithms, and they happen to be right next to each other, that web page will show up earlier rather than later. Okay? And the process by which they do this is very simple. I mean, you could probably come up with that algorithm yourself, and that's exactly what this major search engine implements. Okay? I would argue that an alternate and a lot more rigorous formulation would simply order documents by the statistical significance of keyword occurrence. Okay? So here's a question. You're given a sequence. This is text, plain text. This is the document that you're looking at. And there's a generation model for plain text. So this accounts for, this could be a higher order model. This is not an independent model. This is not an identical model. Preferably some kind of a Markovian model. What is the likelihood of observing a given set of keywords, K1 through Kj, within the shortest subsequence S prime of S of length D or less? In other words, you've searched for average case analysis of algorithms, and you found these within a distance 5 of each other. Okay? What is the statistical significance of this? Well, it turns out the statistical significance of this would be very high. In other words, the p-value of this in the generation model for these words to occur together would actually be very low. And therefore, that document should be posted first on your search results. Okay? It essentially is doing what Google does, what the search engine does in some ad hoc way, but this is a much more rigorous formulation. And we do not know how to do this. If I were to give you a certain set of keywords and ask you what is the p-value of finding these keywords in the distance that you saw in that text, I would not know how to answer this. And I think this would be an awesome result if somebody could show this. All right. A generalization of this goes from sequences to graphs. So I've given you a graph. Here's an example. You can consider your favorite social network, and you can construct a mapping for your node from your node attributes to a scalar. We'll call this the color of a node. For instance, females over 30 making more than $200,000 are mapped to the color red. Males over 30 with 2 children are mapped to color green. Okay? Now, can you argue somehow the statistical significance of tight subgraphs that contain prescribed colors? In other words, do we see an overrepresentation of tight subgraphs containing, for example, red, green, blue? And this could be children, perhaps. And if you can find such subgraphs which are statistically significant, that would tell you perhaps what a family unit is. Okay? So this is just a generalization of what was defined on sequences to graphs. Once again, this problem is hard. All right, so this is something else I mentioned, and I'll give you some results on this. This is arrival sequence of nodes in dynamic networks. So for a dynamic network, how can we infer the likely arrival sequences for nodes? In other words, you're given a graph along with a generation model for this graph. What is the likelihood of observing the graph G for an arrival sequence N1 through Nk? And how do you determine the arrival sequence that maximizes the probability? Okay? Turns out there's a number of neat things you can do. So let's assume there's a preferential attachment graph. Everybody walks in, they're going to connect to M people, selecting probability in proportion to their original degree. Okay? So this is classic preferential attachment. Now it turns out that in such a graph, clearly, if you're looking at the graph after n steps, let's say, there will be one node that came in last, and that has just connected to M nodes. Okay? So clearly that guy has to be the last, but there might be other nodes with degree M as well. Okay? So pick any one of those degree M nodes and you say that that was the guy who came in last. One of two things happens. Either the guy did come in last, in which case by removing him, you created another guy who is now last, or that guy didn't come in last, in which case there's another node with degree M who's still there. Okay? In other words, this gives you a very simple algorithm where you're simply picking nodes of degree M and peeling them off, and you will get to one node. Okay, so I've given you an existence proof that such a sequence happens. Such a sequence is also consistent. In other words, if you replay the sequence backwards, I can actually generate this graph. Okay? Turns out such a sequence is actually also not unique because you can pick any one of those M nodes, and these choices actually are super exponential. Okay? So all I've done is I've defined a DAG with some precedence relation, and I have to search through this DAG, and I have to find that one sequence which is most probable. Okay? Turns out that's incredibly hard because the space, as I mentioned, is actually factorial. It's super exponential, almost done. So how do you optimize within this super exponential space? It turns out that I can actually give you some justification, a theoretical justification, that you don't have to. So here's a graph that shows the posterior probability. So what I've done is I'm looking at the graph, and I've generated these sequences of arrivals. I've generated many, many sequences of arrivals simply by randomizing the choice of which degree M I'm pulling out. Okay? So in this case, I have about 2,000 of these samples that I've shown you. And then once I get a sample, I actually replay the graph. So let's say this node came in first, this node came in second, what is that probability? And then I keep multiplying those probabilities to compute a total probability of what is the probability that an arrival sequence generated that node. Okay? And of course, remember, I want to maximize this probability because I want to get that one sequence which maximizes it. Turns out, all of the probabilities are almost exactly identical. So that's the negative log of probability. And it's almost exactly the same, which means that that probability is highly concentrated. And I can actually almost give you a proof for why that probability is highly concentrated. And the fact that that probability is so highly concentrated tells you that any one of those random sequences that I showed you is about as likely to have led to that graph as any of the other super exponential random sequences that you could have generated from this. So this is a case where essentially you're trying to maximize the p-value, but it's a beautiful case where the hill is so flat and broad that any sequence that you pull is just as good a maximization. All right, so some parting thoughts on these things. I'll argue that significance is essentially is an essential aspect of quantifying the usefulness of an observation. If you don't do this, I don't trust why I should believe that your observation is statistically significant or even correct to begin with. It's an essential component of validating observations. You've seen something. Essentially, you don't have the data to actually make sure that this is correct or not. But if you can give me a p-value that's sufficiently low, I can actually validate that what you're showing me has something of value. Turns out significance testing is hard. It turns out these are really nasty analysis problems that you end up in the middle of, not always tractable. And I would argue that minimizing significance, maximizing significance should be one of the primary goals of what are the algorithmic heuristics that you're applying here. All right. So this is sort of it was a philosophy talk, and really we do, I mean, almost every paper that we write in this area will have significant elements of p-values in almost all the answers that we put there. And actually it works very nicely for us. So thank you very much, and thank you for engaging. And we're standing between lunch.
Somehow this p-value is about you having a model and then you're finding the most surprising observation.
Yes.
And then how does this compare with maximum likelihood where you have a model and then you try to find the
So in fact, that was the last example that I showed you. So in the last example, I have a model, and I'm trying to find an observation that is most consistent with the model. So that was what is the most likely arrival sequence. So
they're actually similar.
they're actually complementary problems in some sense. So in one case, you're given an observation, and you're trying to find out what is the significance of the observation. And in the other case, you don't have the observation, and you're trying to say, what is the observation that is most consistent with my hypotheses? So in some sense, those problems are complementary.
So this is related to Deeparnab's question about if you find something surprising, should you modify your prior or should you? So do people? So one way to get. So whenever we start with the prior, we have some story for justifying the prior.
Yes.
Some kind of generative.
Yeah, exactly.
Principle story
For a non-parametric prior. Yeah.
And then I mean, you could keep asking the sort of an infinite regress question of why that assumption, and so on. Yeah. But at some point, we sort of draw a line and say, this is the story from which we start, and this generates this prior model, and therefore we accept this prior.
So we see this all the time. So invariably, when we try to do this, the first p-values we generate will almost always be either trivial, they will come out high always, or they will be trivially low, they'll come out low always. So the way we end up with these strange priors, the CoDO priors and things like that, I mean, this is an iterative process where you just keep searching for how much more power you want to put into the prior to be able to generate a p-value that has discriminating power. So what we look for the most part, is discriminating power.
Are you sort of inherently searching for a surprising prior? Is that?
That's the converse of the problem. So if you were looking at hypothesis testing, that would be the point of view. And indeed, that is the statistician's point of view. I would almost argue that a computer scientist's point of view is that I'm going to tune my prior to separate out non-surprising from surprising. And if I can find it, more than likely, that is a meaningful prior. It's very unlikely that you will have a non-meaningful prior that gives you classification power. It's so rare for a model to have classification power that if you find it, that prior actually works for you. Please.
And what you mean in practice by classification power, when you're working on one of these specific problems, you're happy if some of the things are significant and others are not.
Absolutely, yes. And of course, it makes sense.
Right, and your sense is that you don't believe you could get into a situation where one prior says this happens and things are real and this happens not.
Yeah.
Another prior says this happens real and this happens not.
Yeah.
In your experience, as a practical matter, that just partly.
In a network analytics case, it almost never happens because that space is so refined that you change one parameter of that prior. I mean, these p-values are almost sigmoidal. You will see something which is a trivial low p-value and a high p-value. There's almost nothing in the middle. And they're so ill-conditioned, the priors are so ill-conditioned that you change one little knob, it'll completely disappear on you.
So your sense is whenever you have divided real data into a non-negligible fraction of yeses and nos, you have also divided it along the right circle. Exactly. And we've done this many times, and I will argue this until somebody shows me otherwise. But it's just designing the p-value. I mean, there are weeks and weeks worth of overnighters that go into designing a p-value like that, where, I mean, it's very simple to say, I'm going to characterize the overlap of two clusters.
by conditioning it on the probability, the density here, the density here, the size here, the size here, the density and the size of the overlap. It's almost trivial to say it after the fact. But to get to that point, there are so many p-values that did not work, which would have no classification at all.
Sorry, Deep, you had a question?
Was there any example in experience where you actually got a prediction of p-values, but when you classified was not scientifically validated?
We don't see that very often. And the reason for this mostly is there's almost very little, so in most of these domains that we work in, there's almost very little direct validation. It's not like somebody's going to tell you that this is a drug molecule and this is not a drug molecule. I mean, that's sort of a zeroth order thing. You want to be able to be right on those things. Most people will then expect you to then say something along the lines of, here's why I believe the rest of these are druggable genes, or these are targets, potential targets. And then the evidence for that is sort of second and third order evidence that you find. Yes, that belongs in a certain pathway, and that's apoptosis, and we know that apoptosis is dysregulated in cancers, and therefore it's right. So we very rarely find those classifications to be wrong. If we have the right prior, and the prior gives us classification, I have not come across a case where the classification was wrong. We have many cases where we don't get good classification. But we don't find it where the classification exists, but the classification is wrong. And maybe there's a clean little result there of why this happens in so many of these network algorithms. And maybe it has something to do with the fact that the space is probably exponential or super exponential. And you're cutting that space in such a way that you have enough samples on one side and the other. And that being the case, you're probably not making the wrong cut. Or at least it's in a family of cuts that's mostly correct.